Hello. Um, I here you are.
Good.
Hi, how are you? Thank you for coming.
Good. Glad to hear from you. Can you hear me well?
Yes, perfect. I can hear you very
Right.
well. Yeah,
So
thank
I
you.
don't know. This seminar will be total to 1 hour. Is that right?
Yeah, yeah, around 1 hour. Is it okay for you? Do you?
Yeah.
We
Sure.
can
Yeah. No
keep it
problem
at
at
the
all.
time. Okay,
Yeah.
perfect. hmm How are you
So
doing?
very good. So what is the format? Can you introduce this clubhouse for me? So because I have never been involved in this activity before. Usually I just go to Zoom meeting or face to face in person.
Yes, of course. Yeah. So this is audio usually, so it was an audio only social media app
Mm.
that became kind of famous during COVID time, or it kind of started during COVID time. And back in time, like back then, it was through invitation only. So there were like a bunch of famous people first, like media people and so on here mostly. And
Mm.
then they grew a lot, actually, I guess, because people like what it's exclusive, I guess, I'm not sure.
Yeah. Mhm.
But also I think it's a different way of social media because you can have conversations that
Yeah. Mhm.
that are more
Yeah.
meaningful, I guess, than just writing short text. Usually Facebook and Twitter and so on, it's just writing
Mhm.
short text, so I think a lot gets missed along the way in the message. And then
So, what are the audience? They're all faculty from NYU.
no, this one is a very mixed audience. So from all over the world, actually, and from
Oh,
different backgrounds. I think that makes it very interesting
I see.
discussion, because it's a very mixed. So they're physicists, then doctors, then scientists like me and you, and then from different fields, engineers like
Oh, got
AI
it.
engineers.
Okay,
So
now I understand. Okay, so I will present about 30 minutes, and the audience might communicate with me. We will discuss about a lot of 30 minutes. Is that right?
yes, exactly. Yeah,
Okay.
if that's okay with you, you can also
Sure.
introduce longer.
Oh,
um
okay. Okay. Now I got it. Mhm.
yeah. So yeah, it's really, it depends on the speaker, and how people want to structure, or how long it takes to explain the
Mhm.
subject from your
Okay.
perspective.
So I
And then,
start
yeah.
from introduce myself, or you will introduce me.
Yeah, I'll introduce you, um,
Okay.
based on
Okay.
on the bio, and
Sure.
and then if you want to add something I didn't say, or
Okay.
or
Okay.
maybe the
Mm.
story about how you got to do this research. Sometimes people think it's also interesting to learn, or if you
Yes.
there some research, talk about how hard or good it was to get a grant for the, whatever you think.
Sure.
The other
Okay.
day we had guest, because she is doing about breaking microbes that became antibiotic resistant. She said it's
Mm.
easy to get grants for the basic science, but then to get grants for translational work, it's very hard, she said. Like, things like that are also interesting to discuss.
Sometimes depends on what field you are in. Yeah. So it's really, for example, Alzheimer's disease. Recently, in the past maybe 4 or 5 years, government spends a lot of money in this aspect. So it's relatively easier to get more funding if you work in Alzheimer's disease. That's why a lot of people used to working on some other diseases. Now they shift their their research focus to Alzheimer's disease. Should we start? Katerina?
Oh, sorry. I
Yeah.
was muted and I didn't
Yeah.
realize. Yes, I was talking and you didn't realize. Yeah, it depends very much on the trend, right? What NIH currently thinks is important and so on. So, yeah. So yeah, let's start.
Sure.
Welcome everyone to the Science Society and a special welcome to our guest speaker. And I hope I'm saying your name right, Keqiang Ye.
Yes. Keqiang
Yes.
Ye. That's correct.
Wow, perfect. I'm so happy. Thank you. And yeah, we he will be presenting his latest really very important research that's quite a breakthrough for Alzheimer's disease. So we are very lucky and I'm very happy to have him. And let me introduce you to give you a little bit of background information. So he is the department chair in the Shenzhen Institute of Advanced Technology in China. And he is the recipient of numerous professional honors, including the Distinguished Scientist Award from the Song Tang Foundation and the American Cancer Research Scholar. And he has published around 220 papers in very like great journals like Cell, Nature, Nature Medicine, Nature Neuroscience, and so on. And his lab is mainly focused on neurotrophin mediated neuronal survival signaling pathways and their role in neurodegenerative disease. He identified numerous novel track A and track B agonists, and these small molecules exhibit potent neurotrophic effect on and display great therapeutic potentials for various neurological diseases, including Alzheimer's disease. Most recently, he found that AEP acts as a novel delta secretase that cleaves both APP and tau and mediates AD pathogenesis. So yeah, he does very innovative research with novel drug targets. So and his lab has identified the small molecular inhibitors that display promising new therapeutic efficacy towards AD. Just to give you a little bit about his educational background, he did his master in science in chemistry, Peking University, Beijing, and his bachelor in science and chemistry, Yilin University in Changchun, China. And his PhD he did at Emory University in Atlanta, Georgia. And his postdoc at the Department of Neuroscience at the Johns Hopkins University. And he was then later assistant professor of pathology and laboratory medicine at Emory University. And now yeah, as I said, he is the department chair and chair professor, faculty of life and health sciences at SIAT. So, welcome. Thank you for taking the time in your very busy schedule, and the stage is all yours.
Okay. Thank you very much, Katerina, for your invitation and kind introduction. I moved from Atlanta to Shenzhen, which is right next to Hong Kong, about more than half years ago. And I have been as a faculty at Emory for more than 20 years. I joined Emory as assistant professor at the end of 2001 and stayed there from assistant to associate to full professor and stayed here totally 20 years. So I just left Emory last summer, so joined this new institute. So I started my neurodegenerative degenerative diseases just by accidentally discovering an enzyme called the AEP, asparagine endopeptidase. Now we have changed the name from AEP into delta secretase because this is the fourth secretase that can cleave APP and promote A beta production. Actually, it also cleaves the base to enhance beta secretase enzymatic activity to promote A beta production as well. So we made this observation in 2014, 2015 to discover that AEP selectively cleaves tau and APP and mediate their role in AD pathogenesis. In 2018, we also discovered a very important transcription factor, CEBV beta, that strongly selectively mediate AEP upregulation in neuron and in the brain. So if we overexpress CEBV beta, it will facilitate. If we knocking down CEBV beta, it will substantially attenuate the AD pathology. So in the past few years, we also have published a bunch of papers regarding CEBV beta. In addition to mediate AEP transcription, it can also mediate ApoE transcription as well. In addition, it will also mediate APP, tau, and the base transcription as well. In the other words, CB beta function as a command-in-chief, mediate all of the major players in AD field, in addition to upregulate AEP transcription and promote its enzymatic activity, which will lead into APP and tau fragmentation, resulting A beta aggregation and hypophosphonated tau form the tangles. So that is the background. So in the past about almost 10 years, we have established CB beta AEP play a major role in driving Alzheimer's disease. Moreover, we have a gradual shift from AD into Parkinson's disease as well, because alpha-synuclein behaves kind of like a tau in neurons. So we have published a series of papers also demonstrate CP beta AEP pathway play a critical role in driving Parkinson's disease onset. So we have also established chronic inflammation driving CB beta AEP pathway will lead to AD onset. Chronic oxidative stress activate the CB beta AEP pathway, which will lead to Parkinson's disease. So it depends on the upstream stress. CB beta AEP pathway activation will lead to different neurodegenerative disease onset. In addition, we have also previously published before, AEP can cut TDP43 and mediate FTLD pathology. So in the other word, we strongly believe CB beta AEP pathway play an essential role in all of neurodegenerative diseases. So in the past few years, we have also extended our study from establish this innovative theory. We have tested different risk factors and their role in driving this pathway's activation to explain why, for example, traumatic brain injury, why ApoE4 or diabetes atherosclerosis can facilitate AD onset and progression. So we have published a lot of paper to demonstrate indeed traumatic brain injury and cholesterol combined with ApoE4 to activate this pathway to trigger AD onset. So now it's a logical extension to our current Nature paper because women are 2 times more susceptible to Alzheimer's disease. This phenomenon has puzzled mankind for decades. So this is our effort to address the hypothesis whether indeed the CB beta AEP pathway also applies to women's susceptibility to Alzheimer's disease. So that's the background. So once we have established this theory to answer why women are more vulnerable will be easier. Um, Previous literature has already published show FSH can activate CEB beta in gonadal cells, driving a lot of reproductive related genes expression. So we just extend previous findings into neurodegenerative disease field. So actually the whole study started from the extended figure 2 and extended figure 3. So actually I will present in a different way, not exactly follow the paper published way, because that was how we made this discovery. So we performed the in vitro experiment first. Actually, in the extended figure 2, we test whether FSH dose dependent or dose dependent to activate CB beta AEP pathway in neuronal cell lines and also in primary culture neurons. So as we expected, indeed, FSH, time-dependent activated CB beta and AEP signaling, driving APP and tau cleavage. And we have also monitored AEP's enzymatic activity escalation, time-dependent. Moreover, the target gene, such as CB beta, AEP, APP, and tau, and also time-dependent being upregulated. The next, we knocking down the receptor FSHR to interrogate whether this hormone indeed through its cognate receptors to mediate this signaling pathway or not. So next in extended figure 3, what we have done is to address whether FSH indeed through CB beta AEP to trigger the downstream APP or tau fragmentation or not. So what we did is in the cell line or primary culture neuron, we deplete CB beta or overexpress the dominant and active AEP mutant, which can block the endogenous AEP. So what we have found indeed depletion CB beta or block AEP, we can successfully blockade FSH-induced APP or tau fragmentation. The next, we just validate by different approaches. In addition to Western blot, we're also using immunofluorescence to validate different downstream biochemical events. So in the same figure, in the extended figure 3, we have also demonstrated indeed this signaling is through its receptor, FSR, which is a G protein coupled receptor, which can activate the downstream ERK signaling, which can lead into CB beta activation through phosphorylation. On the other hand, the AKT is also activated by the receptor, which can lead into SRPK2 phosphorylation, which is the upstream kinase for AEP. We have published a molecular cell paper in 2017. We demonstrate SRPK2 phosphorylation of AEP can drive AEP translocation from lysosome to the cytoplasm where it can cut its substrate. So and then we we show indeed FSH through the FSHR and by activating downstream ERK and AKT, which then through phosphorylating SRPK2, then subsequently can phosphorylate AEP, activate AEP. So we also use a ERK inhibitor or AKT inhibitor to further validate indeed antagonize those kinases. We can block the FSH induced CB beta AEP activation. So those are the in vitro experiment. We validate indeed FSH can through its receptor to activate this CB beta AEP pathway leading to APP and tau fragmentation, resulting A beta or tau pathology. So next we extend to the figure 4. Actually, that's the first experiment that we perform, because this is the most important experiment. We want to do the most important experiment first. So we using young 3xTG male and female mice, and then we inject the recombinant FSH into this animal. We ask whether we can facilitate AD pathology on site earlier or not. So in figure 4 and also extended figure 5, 6, 7, we are addressing these questions. So in figure 4, let me see, in figure 4, we also monitored the biochemical event after a few months FSH administration. And as we expected, indeed, AEP is upregulated, which leading to APP and tau fragmentation. And also we performed the behavioral experiment as we expected, indeed, as the APP and tau fragmentation, the cognitive function is impaired. In the extended figure 5, 6, and 7, actually, which validate all the downstream pathology effect, such as we performed the beta immunohistochemistry, soil staining, to demonstrate indeed FSH can enhance beta or tau aggregation. Of course, we also validate human recombinant FSH concentration, as we expect an increase in the brain, but the LH, another hormone, was not altered at all. And moreover, the downstream target, CB beta, AEP, APP, and tau expression significantly increased. So we also perform gauge staining and perform the EM to demonstrate the dendritic spine and also synapse reduction after FSH treatment. Next, we also performed a bunch of immunofluorescence staining to further validate the biochemical observation we made by Western blot. So those are just different approach to validate the similar observation. Next, in the extended figure 6, we supplement with estradiol. The reason we supplement E2 is because FSH can repress the estrogen or estradiol's concentration. So we want to exclude the potential role of estrogen. So that's why we put it back to maintain estradiol's concentration in the presence of exogenous FSH. So we found, again, FSH can trigger AD pathology even in the presence of the estradiol. So this study suggests FSH trigger radionuclide independent of estrogen. So pathogenesis is unrelated to estrogen reduction after menopause. So this why we perform this experiment. So in extended figure 7, just demonstrated the pathology and biochemical experiment again. So all of this experiment just want to demonstrate one conclusion. FSH indeed in vitro and in vivo activate CB beta and drive ready onset independent of estrogen reduction after menopause. So next we move to figure 1 and also extended figure 1. In this figure, we try to use different model. Usually for the ovarian failure, people use ovarectomy as a surgery approach to remove ovarian to mimic the menopause. This is the extensive employee the experimental model to mimic ovarian failure. So we use the same model. We're using the young female 3xTG mouse model to remove the ovarian. And at the meantime, we're also using anti-FSH antibody to specific neutralize the upregulated FSH hormone. We ask whether we can reverse or alleviate FSH induced AD pathology or cognitive deficit. So basically, in figure 1 and extended figure 1, we using different approach to demonstrate indeed, as we expected, FSH antibody successfully, strongly diminish Western blot staining, demonstrate support this observation, and we also perform Morris Watermaze to demonstrate neutralize FSH, indeed alleviate overrectomy induced AD pathology and cognitive deficit. Moreover, we have validate using different animal model. In addition to 3xTG, we also use the male APP-PS1 mice by injecting the FSH. We can demonstrate the similar effect, A beta production increase. Next, we move to the figure 2. In figure 2, actually, we want to answer whether the receptor FSHR indeed expressed in neuron in the brain or not, because previous study always focus on the gonadal cells or granulosa cells about the FSR mediated reproductive related signaling transductions. So we shift our study about this signaling to the neurons. So that's why we use extensive approach to demonstrate this. So what we did is using human cell lines or human tissue or mouse tissues. Here we use the cortex, hippocampus, or warfarin as a positive control, and also use rat neurons. Anyway, we use a different tissue to demonstrate that indeed FSR is expressed in the central nervous system. And we also demonstrate indeed if we knock out FSR receptor, immunoblotting observation will be abolished. We also conduct immunohistochemistry stainings using the testes as a positive control. Here is a knockout mice. Of course, the receptor is completely diminished from the testes. Again, in the dentate gyrus or anterenal cortex, the expression was also being abolished. So next, we're using immunofluorescent staining to demonstrate the knocking down FSR in the neurons. We can indeed validate or its different cell types distribution. So this study is to validate, confirm indeed FSR expressed in neurons in the central nervous system. So next, we extend our study into the in vivo using the FSR knockout mice. Or no, we use the siRNA to knocking down FSHR in the 3X TG's hippocampus by injecting the virus to deplete the receptor, and then use the overreactive and the CB beta AEP signaling pathway will be repressed, which will leading to APP tau fragmentation reduction. So it can block the overrectment induced the AD pathology. So those has been demonstrated in extended figure 4 in addition to figure 3. So here we demonstrate the FSR deletion in the hippocampus can attenuate OVX induced AD pathology and cognitive decline. So next we extend our study into figure 4, where we're using the recombinant FSH, which I have talking about before. In addition, we addressed whether this hormone can induce any pathology change in wild-type mice or human APP lucky mice. The reason we perform this experiment is we want to know in under baseline or without any exogenous overexpression human APP or tau, whether upregulation of FSH is sufficient to induce the AD pathology or cognitive deficit or not. So in the extended figure 8, actually here we using the wild-type mice and human APP lucky mice. We found out, indeed, as we expected, exogenous recombinant FSH administration will lead to CB beta upregulation and the AEP increase, which will trigger APP and tau fragmentation. However, in wild-type mice, we do not see A beta aggregation. By contrast, in human APP lucky mice, we do observe the protein inclusions in the hippocampus demonstrated by silver staining. So indicating presumably in wild-type mice, the mouse A beta are reluctant to aggregate it. Those has been proposed before. By contrast, human A beta are more prone to aggregate it. So that's why we can say the human APP, not in mice, in response to FSH treatment, the silver stain result in the hippocampus. In alignment with this A beta upregulation, behavioral studies suggest indeed human APP not in mice display the cognitive defects By contrast, the wild-type mice do not show any memory disorders. Next, we also use an immunofluorescent approach to demonstrate indeed in human lucky mice, FSH treatment can activate APP fragmentation and also neuronal cell death and also A-beta upregulation. So next, we further to interrogate whether FSH induced AD pathology and behavioral deficit indeed through CB beta AEP pathway or not. So next, we cross 3xTG homozygous knockout mice are embryonic lethal, or some of them even survive. However, they have some other uh, defects because CB beta play a critical role in adipocyte differentiation. So that's why we did not choose the homozygous mice. We only use heterozygous mice to uh, breed with 3xTG. So we in figure 5, we after we obtain this 3xTG beta heterozygous mice, then we use two different models to further investigate whether indeed the FSH can trigger CB beta AEP mediate APP and tau fragmentation, leading to the AT pathology and cognitive impairment. So, as we expected, indeed, knocking down or partially delete CB beta can robustly repress CB beta AEP pathway, leading to the reduction of APP and tau fragmentation. Consequently, the cognitive deficit is alleviated. Moreover, we use the overrectomy as an alternative approach to further ascertain indeed overrectomy induced AD pathology and behavioral deficit can be alleviated when CB beta is partially depleted. And the pathology and the biochemical evidence are accumulated, are demonstrated in the extended figure 9 and figure 10, where we documented FSH induced AD pathology by immunohistochemistry staining and immunofluorescence staining, and also we used dendritic spine staining, gauge staining. All of those demonstrate indeed depletion of CB beta can significantly alleviate FSH induced those pathological effect. In extended figure 10, we made a similar observation, but here we used overrectomy as a model to drive AD pathology. And once again, depletion CB beta can significantly attenuate overrectomy-induced AD pathology and cognitive deficit. So, in a summary, so in all of this data, we successfully demonstrate no matter using overrectomy as a model or exogenous recombinant FSH administration can induce CB beta AEP pathway activation, which will subsequently cleave the upregulated APP and tau into the fragment. We have previous demonstrated before this fragmentation will lead to A beta transconation and the neurofibrillary tangle formation. So using the FSH antibody, we can specifically neutralize the recommend induced upregulation of FSH to abolish overreckoning triggered onset and progression. Moreover, we have provide extensive and knockout FSHR can also abolish overrectomy-induced AD pathology and cognitive deficit. Next, we also provide in vivo evidence to further demonstrate indeed FSH or overrectomy through CB beta AEP pathway in vivo to mediate the AD pathogenesis and learning memory defects. So that's all. Katerina?
Yes, thank you so much for explaining this paper so well to us. So please flash your mics if you have a question. Raise your hand in the audience, and yeah, let us know that way who has a question first. I saw Catherine. You have a question. Go ahead.
It's a bit more of a comment, and I do have a bit of a question as well. So Lisa Mosconi's group and Richard Isaacson and Cornell, they recently published about how important hormone replacement therapy may be estradiol for protection of cognitive decline. And this certainly follows, as we know, also the North American Menopause Society has guidelines recommending hormone replacement therapy for women, but we don't really recommend by the guidelines to be checking FSH and LH levels. But I can tell you in my practice, I check them and we try to keep them, in an early menopausal time. Some levels can go as high as 100. Most women, FSH around perimenopause will be 25 to 60. And so I think it's interesting to see this data and to think where my question would be, are you 100% blocking with the FSH antibody treatment? Are you getting serum levels? I didn't see that. And do you think that would be something to correlate? Thank you.
Okay, perfect. That's a wonderful question. Indeed, the hormone replacement therapy have been previously tested in many human clinical trials, but a conflicting observation have been reported. Some of the estrogen supplement show some beneficial effect in AD patient, but many of the hormone replacement therapy show either no any beneficial effect or sometimes even have a adverse effect. So it's controversial whether hormone replacement therapy play or can help to alleviate AD patient's symptom or not is inconclusive yet. So our study suggests that the antibody, which is a specific targeted beta subunit, can successfully neutralize FSH hormone. Those has been demonstrated extensively in our experiment. We have provided a bunch of evidence to demonstrate indeed our antibody successfully penetrated into the brain where it neutralizes the hormone without interfere with another hormone, LH, and can successfully block the FSR-mediated signaling transduction. So you answer your question, indeed, this antibody is very powerful, which has also been shown before, anti-FSHR can increase the bone density, because after menopause, the osteoporosis will be taking place in female, and also the visceral adiposis will happen as well. So this antibody has been shown by Dr. Mon Zaidi. Anti-FSHR can effectively treat osteoporosis and the adiposis through the upregulation the energy expenditures. So in this study, he conducted extensive study using this antibody to validate some of the concern you just raised. Actually, now we have just got the joint grant. So we are initiating the IND enabling study to address whether anti-FSHR beta antibody being used for treating Alzheimer's disease. So this IND enabling study with the humanized antibody just started. Next question.
Yeah, please go ahead and ask the next question. Please flash your mics if you have a question. Yep, Serena Wei, go ahead.
Yeah, just a point. What we currently know in terms of the neurotoxicity of the beta amyloid plaques versus smaller tetramers or octamers. At one point, we were starting to get some evidence that the tetramers or so were more specifically toxic. Could you comment on what we currently know?
Because I missed your first part of your question, because I guess the signal was not quite clear, not stable. Can you repeat
Sure.
your question again?
Sure. Can you hear me okay now?
Yeah, I can hear you fine.
Okay. I know the question was about the specific toxicity of the beta amyloid plaques versus smaller tetramers and octamers of the beta amyloid. At one point, there was some evidence accumulating that it was the smaller tetramers were much more neurotoxic. They'd poke holes in the membrane and so forth. I'm just curious if you could comment on what we currently know about the specific neurotoxicity and the forms of the beta amyloid that are most toxic.
I got your question. So you are asking whether the A beta toxicity is come from the tetramer or different oligomers instead of the
it's the beta amyloid plaques themselves versus smaller fragments of the plaques. Am I cutting out again?
No, I think this time, Doctor, I think you're cutting out. Maybe your connection is not good. Oh, now you're muted. I see that you're muted.
Are you able to hear us, Dr. Wei?
Oh, you're asking me? Yes, I can hear you.
Okay. Dr. Ye, the unmute button, I don't know if can you hear us? Wait, let me try to message him.
Are you there?
Oh, yes.
Yeah.
Yes.
Oh,
Can you hear
okay.
us now?
Can you hear us?
Yeah, now I can hear you. Sorry. Yeah. To
don't
answer
worry.
your question, in the field, the consensus view is indeed is A beta oligomers are more toxic, at least in vitro experiment or some of the in vivo evidence, and people believes the A beta deposit into the cell plaque is kind of protective because it can sequester the oligomers of A beta into the depot to prevent the diffused A beta oligomers to contact the cellular part. So, for example, some of the receptors, such as NMDA receptors on neurons, so they can prevent those A beta oligomers to exert their toxic effect. So that's the consensus conclusion about the A beta oligomers neurotoxicity. Any other question?
I think Dr. Wei, did you unmute before to ask a question? Okay. Varun, did you have a question?
Oh, nothing actually. I was just going through this article that you have pinned against the wall. Let me just go through it once. Then I'll just chime in.
Oh, sure. Dr. Maria, you have a question?
Yes, I do. Thank you, Doctor. I don't want to mispronounce your
Dr.
name.
Yi. Yeah, Yi.
Yi.
Yes,
Yes, Dr. Yi. Thank you for your presentation. That was really interesting.
thank
So
you.
I was just wondering because I'm not very familiar with the SHSY5Y cells.
Mm.
And I can see in one of your figures you measured the relative IL-6 and IL-1 beta. And
Mm.
I was wondering if you had done any experiments post blocking the FSH, if that reverted the cytokine levels back to match the control levels. I hope my question makes sense.
Yes. So you are asking, first question is what is the SHSY5Y cells, and also why the FSH treatment can enhance those inflammatory cytokines, whether we block FSH can decrease those inflammatory cytokines. Is that right?
Yes, correct. Thank you.
Okay, so sure. The SHSY5Y is a human neuronal cell lines. Those are frequently being used in many studies. If you do not want to use a primary culture or to do some preliminary studies, so that's why here we use two cellular model. One is a human SHSY5Y, those neuroblastoma cell lines. Next, we also use the primal culture neurons to demonstrate indeed FSH through its receptors to upregulate the signalings. So in vivo, indeed, we administration of FSH can activate the CEBP beta, these pathways, because this transcription factor, CEBP beta, can not only being activated by inflammatory cytokine. This transcription factor is very interesting. It can function as a transcription factor for many cytokines, such as IL-6, and it has been published before. Say, CB beta's original name is ILTF, interleukin 6 transcription factor. So this transcription factor can not only mediate the inflammatory cytokines expression On the other hand, it can also be activated by those cytokines in addition to be activated by A beta. So to neutralize the FSH by its antibody, we can successfully to decrease those inflammatory cytokines because CB beta activation is being blunted. So any other questions?
Hello, hi, Dr.
Hi.
Ye. Uh,
Yeah, I
this
can
is
hear you.
Wei from NIH. That's a fascinating story. So I am curious, what do you think the normal function of FSH in neurons?
That's a very good questions. The short answer is I do not know. Yeah, I do not know because in the field, people over the past decades just focus on this FSH, FSHR signaling in the reproductive system in the gonadal cells. And so this is a very few experiment demonstrate indeed this signaling can play some role in central nervous system in neurons. So the short answer is I do not know physiologically what do they do. Um, so in the future, maybe we could explore in this direction.
Thanks. But do you know how broadly are they expressed in neurons? Do they specifically in the hippocampal area or across the whole brain? All the
Uh,
neurons express the receptor?
Experiment has been addressed by Dr. Zaidi's group because his field is endocrinology. His whole research is focused on the FSH, FSHR signaling. So that's why we collaborated. My focus is mainly is the Alzheimer's disease, CB, beta, AEP pathways in AD and PD. So that's why we collaborated. So he has done extensive study. In addition to the hippocampus, such as dentagerous, he also demonstrated this receptor expressed in the entorhinal cortex. And also he has shown this receptor can be expressed in other brain region in different neurons. However, he did not observe this receptor, at least not in high abundance, in astrocyte or microglia, so mainly expressed in neurons.
Mm.
However, we do not know physiologically what does FSHR do in neuron.
Thank you.
Sure, you're welcome. Any more questions?
Yes, Dr. Yi, thank you so much. I just want
You're
to
welcome.
just want to double check. So this has clinical application for men as well as women, no?
You are talking about the CB beta AEP pathway or talking about FSH?
Talking about the FSH blockade.
Oh, okay. Since FSH play much more important role in female than male, I'm talking about for the lady onset. Even though we're using both male and female 3xTG mice, exogenously administrative of a high dose of FSH to mimic the after menopause escalation of FSH. We do observe the AD pathology uh, happening in both male and female mice. However, it's our belief this clinical trial will be mainly focused on the female patient. Presumably, we might in the future choose some male patient. However, that's not what we believe. It's really due to FSH increase in male. Even though it's increased a little bit, over the time. However, in female, is increase 30, 40, or even 50 folds. In male, only 2 or 3 folds during the aging process. So that's why we believe the antibody might be more beneficial for the female patient as compared to the male patient.
Yeah, doctor, what are the symptoms of Alzheimer's diseases other than loss of memory?
Can you speak a little bit?
Yeah, I was asking you what are the symptoms of Alzheimer's disease other than loss of memory? Hello? Hello? Am I audible? Hello?
Hello, Varun. We can hear you. I don't know if Dr. Yi is able to hear you.
I'm still, so I couldn't hear well.
Oh, yeah. Now we can hear you again.
Doctor, my question was predominantly based on whether your memory loss is like short term or like do you like forget and you can recollect or like I mean if you forget things like they are gone permanently. How is it? Hello?
I still cannot hear the question well. Katerina, can you help me so that I understand the question better
Yeah.
because the signal is not quite stable.
Varun, you're asking what the other symptoms of Alzheimer's is, or what the other
Yeah, both
function?
both question. Yeah, both question. But when they say memory loss, right? So I mean, how would you actually define memory loss? Like, what is actually happening when they say you're losing memory? I mean,
So what are
are
the
you like,
clinical indicators of the result? Like you're asking for the clinical?
no, no, not clinical. I'm asking something in general, whatever it is. Uh,
What
Generally, how would
Alzheimer's
you?
in the cells, in the brain cells? What happens?
Yeah, yeah, Alzheimer's in the brain cells.
Yeah. Do you want to answer, Dr. Yi, or should
So
I?
let me first understand his questions.
Oh, he's just asking in general what happens in the cell when Alzheimer's occurs. Like
So he's
what?
asking the general.
Yeah.
Oh, okay. Now I got it. Okay. Actually, there's no short answer for this question because this Alzheimer's disease is a very complicated process and it takes about 20 or 30 years to happen. So usually, for example, A beta start from our full, the forehead at the beginning, at the later 30 or earlier 40 years old. Okay, tau happened in the brainstem a little bit later. So these two pathology happened in our brain at a different time and at different locations. So our memory is mainly mediated by the cholinergic neurons in the hippocampus. So when Alzheimer's happens, many of these cells responsible for uh, memorize or store our memory start to degenerate. Mainly is at the beginning is the basal forebrain cholinergic neurons. Those neurons will be very vulnerable for A beta or tau stress. So at the beginning, those neurons start to die, and then at that time we also have a cumulative inflammation going on in our brain because there's a lot of insoluble aggregates such as A beta or insoluble tau. They will widespread in our brain. They will trigger a lot of inflammation. The chronic inflammation will amplify the signaling, what I just talking about. And those could be exacerbated after menopause because the highly increased FSH could activate the signaling I just talking about, to leading to more and more A beta production or tau fragmentation. They will aggregate into the tangles or insoluble inclusions, will further trigger the inflammation. Those will leading to more and massive neuronal cell death, and will leading to our memory loss. So that's the whole picture of what happening in our brain over the, let's say, start from later 40 or earlier 50s until maybe 70 or 80s or even later. So those will last about 30 or 40 years, this whole process, actually. Yeah, so it's a very complicated process. Cannot be, there's no any short answer to address your question. Any other scientific questions?
Yeah, just to follow up Catherine's comments, questions about hormone replacement therapy, specifically estradiol. I didn't quite follow what was inconclusive about whether it helped, but could you summarize any indications of how estradiol supplementation could somehow help, and what was inconclusive about that?
So let me, because the signal was unstable, I did not hear well, but I guess I catch a part of what you're talking about. What is the inconclusive about the HRT in clinical trial? Is that right?
Yeah, and specifically estradiol, yes.
Yeah. So for those hormone therapies, as I just indicated, there's numerous clinical trials have been performed in different centers. There's a lot of publications in this field. As I just talking about, there's conflicting observations. Some reports say it's beneficial. Some say it's no any effect at all. Some even have a adverse effect because no matter estrogen or estradiol have been demonstrated they have the potential or have the risk to trigger the cancers. So that's why people even worry about using the hormone therapy for treating AD might induce some cancers. So that's why, in addition to one of the concern.
Yeah, he
Yeah,
was a little bit in the matrix, but Serena, if you'd like, I can tell you I'm certified in menopause care, and the data, I would call it dirty. It's conflicted. We have data that includes synthetic estradiol, like Premarin, Prempro. That's the Women's Health Initiative data, and it showed a signal for perhaps increased cardiovascular events, increased incidence of breast cancer. Now, when that data is a little cleaner and we're using less synthetic estrogens, and we're looking at cohorts of women who don't otherwise have diabetes, obesity, smoking, it's hard to really see because we're also seeing the aging, and how much is aging contributed by
Yeah,
that lack of estradiol and that heightening of FSH, which is a signal that we see in perimenopause as well. And women will report very commonly in the perimenopausal and menopausal transition brain fog, confusion, forgetfulness. And the goal is to replace estrogen in the lowest dose for the shortest amount of time possible with the safest estradiol in an appropriate candidate. So a woman who doesn't have a known risk of cancer or active cancer, doesn't otherwise have a stroke or cardiovascular profile that would be worrisome. But estradiol itself tends to be very anti-inflammatory. It confers cardiovascular protection in many ways. We see less atherogenic cholesterol particles in a woman who is premenopausal. So I would say this contradictory data is not surprising because it's very difficult to look at this. And I think ideally having the appropriate candidate who's otherwise has optimal health parameters is really the goal, and something we should be looking at all of the factors that contribute to a woman's vascular health and not just the hormones.
Oh, thank you very much for the explanation. Yeah, I learned a lot. I appreciate.
Doctor, could you explain about this sequence? That LVYKDPARPNTQZ. If you were to go to the article, the first page, FSHAB prevents the AD phenotype. Second paragraph.
Katerina, are you there?
Yes, I'm there. I'm trying to look at what.
First page, second paragraph. FSHAB prevents the AD phenotype. In that,
Yes.
uh,
What is unclear?
can you throw some light on what does that sequence mean? Amino acid mouse FSH beta sequence
Oh, oh,
LVYKDPARPNTQ.
okay. So this is the antigen we Dr. Zaidi used to generate this antibody, so that this anti-FSH beta antibody can survive.
Dr. Wee, I think you're breaking up again. Can you hear me?
No, I think he's muted. Dr. Ye, you're muted. Hello?
Can you hear us? No, I think you're still muted. Yes. Can you hear us now? We can't hear you. Maybe we can talk. I think it might. We could hear a little bit, but you're still breaking up. Maybe it's a connection issue. If you maybe turn the Wi-Fi on and off, maybe it improves. Or I don't know. You're muted again. Can you wait? Can you write in the chat if you can hear us or not? I don't think Dr. E can hear us currently. So I'm not sure what he was explaining exactly. But
Well,
I
it's okay.
think
I think we can
about
move
the
on with the next.
antibody. I think it's the antibody they use to basically block the hormone.
Yeah.
Please. Oh, now we can hear you.
Because the signal was unstable. I'm sorry. Yeah, I I missed it. So I'm not sure whether I have answered that gentleman clearly. Just in case he did not hear it, I will repeat it again. So that sequence was the antigen we used to generate the antibody. So Dr. Zaidi used this antigen to make a specific antibody. This antibody can selectively bind to the hormone prevent the hormone binds to its receptor to neutralize the signaling. So this antibody does not affect the concentration of the hormone in the, only block it from bind to its receptor to exert the biological function.
I have a question. Are you currently planning to do clinical studies with the using the same approach as you used in the mice?
Yes, as I just indicated, Dr. Zaidi and I, and the Emory Group got the NIH funding to do the IND enabling study before we do the clinical trial. So if we demonstrate indeed this humanized antibody are safe and also can be manufactured in large quantity and demonstrate indeed we could reproduce this observation in independent hands, then I guess the clinical trial will be initiated maybe in 2 years.
Oh, that's wonderful news. I hope
In
everything
addition to
goes.
that, actually, we have the AEP inhibitors, and those can be more extensive used in addition to AD and also for PD, for Parkinson's disease, and also can be used for even treated osteoporosis as well. So the inhibitors, that drug against AEP now is in the pre-IND stage. So the clinical trial could be also initiated maybe in a couple of years as well. Yeah, that's a different alternative approach to target this pathway.
Yeah, that's I wish you all the best for
Thank
the
you very
next
much.
year. I think it will be very important, and I will be in line at some point to take it.
I hope
Because,
our medicines will be helpful for you.
yeah, for all of us. I mean,
Okay,
you
sure.
know, just in case, I would just take it. Um,
Okay.
but
Any more questions in the
yeah,
audience?
exactly.
Yes, yes, Dr. Ye. So I was wondering if there's any epidemiology data for women that went through ovarectomy. Do they have a higher incidence of Alzheimer's disease?
Yes, those actually was documented in our original manuscript, and
Mm,
because nature, the world limits, I guess maybe some of those literature been removed. Yes, the short answer is yes. There's a tons of epidemiology study demonstrate the overrectomy surgery will enhance the AD risk in many women.
cool. Thank you.
Sure.
I have another question with respect to ApoE allele.
Mm.
Are there any studies looking to see if there's, you know, interest in looking at the ApoE 3, 4, or 4, 4 with?
Uh-huh. Oh,
Mhm.
good question. Yes, those actually has been extended study in literatures for female. One copy of ApoE4, they uh, are prone to develop AD much earlier than male. However, for ApoE4, those are homozygous female, and they display the similar pace as the male. So in the other word, single copy of ApoE4 compared to men, female are more vulnerable. Those has been published before in human. Yeah, there's a lot of clinical study demonstrate that. And we have a research paper to addressing why ApoE4 could exacerbate FSH effect in triggering the AD pathogenesis.
I have a question. Could FSH have a similar? I know the levels in men doesn't change as much, but could there be a link to Parkinson in male, and that in female for whatever reason it
Uh,
triggers more Alzheimer's, and then in male maybe Parkinson?
Oh, that's a wonderful question. Actually, Parkinson's disease is relatively more abundant in male compared to female. AD is opposite. In AD, female is 2 or 3 times more for women than men. Parkinson's disease a little bit higher for men. There's no any evidence to suggest FSH might play any role in Parkinson disease. Actually, in my lab, we are starting to explore this possibility. However, it's too premature to answer whether it play any role in male Parkinson disease by this hormone or not.
Oh, thank you. Yeah, I'm curious to hear.
That's a
Yeah,
wonderful question. Yeah.
thank you.
Yeah, and of note, hypogonadal men do have higher levels of FSH. They don't get as high as we see in women in menopause, but they certainly can double out of normal range for a man who
Mm,
has lower than optimal free testosterone levels. So that is a biomarker that we use when considering replacement, because for men, they make estradiol from testosterone. So there is some protective features, we believe, of estrogen and where the cardiovascular risk can lie for men who are testosterone deficient.
mm, mm. Thank you. That's very informative.
So related to that question regarding female male difference. So are you indicate that the pathophysiology of Alzheimer might be different between male and female? For instance, if male has much lower level of FSH, does that imply the neuron may express higher receptor or sensitivity is different? Or what do you think? What are your thoughts on this difference between male and female, particularly in the whatever happens to the neuron part?
Okay. So we do not think FSHR expression there's any difference in male or female neurons. For those cells, they are the same. Yeah, the expression abundance or level might be comparable. It is the hormone that drives the signaling activation is hugely different between the male and the female, which leading to the downstream signaling highly upregulation in the female compared to the male. So it's not the receptor distribution or expression level difference in neurons.
I, I see, but the difference level of FSH is, as you mentioned, alluded, hugely different
Yeah.
between in women is like maybe more than 10 times higher. So how do you envision for men to develop Alzheimer? What because
Okay.
yeah.
That's a good question. So for the risk factor for men, mainly is for example, diabetes, atherosclerosis, and hypertension. Those has
Mm.
been well documented. Those age-related chronic diseases are high risk factor for male to develop AD in addition to traumatic brain injury. Those diabetes, atherosclerosis, and hypertension, they are the risk factors. And also, as we just indicated, ApoE4, this gene can also highly increase the possibility of AD onset and progression for male as well.
So are those risk factors in any way related to the FSH signaling
No,
at all?
those are independent. So that's why male can also develop AD as well, just because they have those hypertension, diabetes, or atherosclerosis, or ApoE4 genes.
Okay, thank you.
Yeah, FSH just give additional risk factor to increase women's odds.
Thanks.
Sure. Okay, Katerina?
Yes,
If,
thank you.
yeah, yeah. If no more question, then I gotta go. At 10:30, I might have some other meetings going on. So
Yes,
thank you very much for the opportunity. Maybe if there's more questions, you can email me. I could answer it through the email.
yes, thank you so much for taking the time and for staying longer than we planned. There were a lot of questions, and we couldn't go.
Sure. Yeah, you can forward me. Yeah, through the email. Yeah.
Yes, thank you so much again. It was
No
very
problem.
awesome.
You're welcome. Okay, it's really nice to talking to you and the audience. It's really my great pleasure.
Thank
Okay,
you
I
so
gotta go.
much, Dr. Ye.
Okay,
Thank you so much.
Bye.
Really
Thank you.
Thank
appreciate
you,
it.
Dr. Yi.
yeah. Okay.
Bye.
Bye
Thank you,
bye.
Dr. Yi. Bye.
So much.
Bye. Very nice. Thank you.
I had a follow-up question, though. And Catherine, I back-channeled you. I'm wondering if it's in terms of the HRT and the whether progesterone it has. I was going to ask Dr. Yi if progesterone had been looked at, but I understand that in combination with estradiol, it can have some protective effects against some of the cancers. I don't know if anyone has any further information on that.
Yeah, we use progesterone in women who have a uterus. You have to use progesterone because it does offset the proliferative changes that we see in the endometrial lining of the uterus, which can predispose women to endometrial cancer. So you have estrogen receptors in the uterus, and women can have dysfunctional bleeding when those cells become proliferated with hormone replacement therapy. It's quite uncommon, but there are some women who have genetic risks for that, and there are some women who are just very sensitive to estrogen and can get some hyperplastic cell changes there that can go on to be carcinogenic. So the good news is those cancers tend to have symptoms, if you have that complication of hormone replacement therapy, because you will have vaginal bleeding. Progesterone otherwise has uh, neuroprotective effects in the brain. It's a precursor to allopregnanolone, which is a GABA pathway, and so it can be calming. It's an anxiolytic. It also offsets some of the bloating. It's a mild diuretic, so it may work on some vascular components as well.
Does it offer any protective effects towards breast cancer?
We don't have great data on that. They stain breast cancers for estrogen and progesterone receptors, but of course we know there are other factors at play for women's breast cancer. So progesterone tends to offset the bloating that can come, so there can be more cystic change or density in the breast related to progesterone use. That's the extent to which I can answer your question.
Thank you. Thank you.
Yeah. If so, as Dr. Yi said, if you have questions, please back channel them to me or contact me, and I will forward them and answer them. So yeah, if you want to continue the discussion, it's fine. Like we can keep the room open and continue the discussion. Yeah, let me know because Jake, I know you came to the stage, and Leslie, nice to see you, Leslie. You came up. Maybe you wanted to comment or share something. Please go ahead.
I got a question, if possible. Is now the good time to ask it?
Yeah, I hope I can answer it. If not, I'll forward the question and then get back to you.
Okay. I got a buddy who's got Alzheimer's, and I wonder. I'm very. This is all new to me. I've just sort of been exposed to it. I'm curious as to why people that have Alzheimer's have it, and then others that don't have it don't. The question is how when you go to sleep, your brain is sort of cleaning itself out. There's a major chemical change inside the brain itself to sort of like purify it. Is this sort of a disorder because there's something wrong with the cleaning process, or is it other than that?
Yeah, so the paper we talked about, or Dr. Yi, his paper, his really extensive study, this was more the case for the higher risk in women and Alzheimer is that this hormone, he showed direct evidence that this hormone um, can elicit ADD, and by blocking it in different ways, he blocked it with an antibody, and then he made knockdown versions, and then he took the ovaries out in the animals. So he showed it in very different ways that this hormone can elicit basically Alzheimer's. But in men, he said that there are other mechanisms that most likely trigger this. And yeah, you talked about that the brain is not efficient anymore at cleaning out proteins that are misfolded and so on. And a big role there play the glia cells. And yes, it has been shown that chronic inflammation, and if glia don't do the job anymore of cleaning out, of cleaning up basically, then there's a high increased risk for dementia or Alzheimer's, for having these plaques. So yeah, and sleeping, having enough sleep plays a huge role. Hey Leslie, you want to say something?
Oh no, I'll catch him later because I had some specific stuff with Parkinson's, but no, I just had a comment because it's not my field of specialty, but I just caught a study show, with reference to the sleeping. It was really interesting. They showed a correlation both ways between napping for elderly people developing Alzheimer's, and then in reverse, Alzheimer's patients do more midday napping. So that tangentially might play into some of the sleep issues because elderly people tend to have shorter amounts of night sleep and not as deep.
Yeah, that's oh, thank you for reminding me, Leslie. And there came another paper out last week, or was it this week, that showed that there are specific genes, like they found that in some people they have, I think, 4 or 5 genes that are responsible for having a really good quality of sleep, even if it's just a few hours. And in those people, they are way more resilient in the way that they don't get mental health issues and Alzheimer's, dementia, and Parkinson. They are protected against that. So the amount of sleep apparently is depending on what genetics you have. So the amount of sleep is not necessarily the most important factor. Is the quality of sleep in the time you sleep. And yeah, so genetics apparently plays a bigger role than we thought before.
Yeah, that would be actually interesting to look at any correlations between bipolar and both Parkinson's and Alzheimer's, as far as brain clearing, because bipolar is mainly a circadian, well, it's a big part of it is a circadian rhythm failure, basically.
Yeah, also autism, it's a, also an issue of being able to sleep well. I think one big factor is uh, um, the zinc factor that influences then the melatonin release, and then affects also the sleep quality, and then also vitamin D levels, I think. So I have to look back into it at some point. I wrote like a short review about it, but it's so many years ago. I'd have to look at, but there's a, yeah, so hopefully we find ways with these new studies how to increase in people quality of sleep, maybe block this hormone. I'm really looking forward to have them a human way of blocking this hormone, hopefully in the next 10 years for me. I'll just take it. I mean, there's doesn't seem to be anything bad about it. Your bone density stays healthy. You don't get so fat. Your cholesterol, and you're protected against Alzheimer's. I don't see any downside with this. Really rare.
Yeah, I would also like to comment that I'm not really convinced that Parkinson's is, especially when you look at all the Parkinsonians diseases, that there's really a sex-linked bias, because there's a huge societal bias once we get to doing research with Parkinsonians patients. Unfortunately, our cohorts are typically 85 to 90% male. That is because they have they typically, the people typically have a woman at home that is a spouse at home that is supporting them to come in and through the complexities of research. While we don't find this quite the same thing in reverse. Now that we're looking more at prodromal, well, this actually plays into it, because uh, we're looking back to essentially sleep disorders, as far as not if you don't inhibit muscular motion. That's really easy to catch in a sleep study. For a full diagnosis, you also have to act out dreams, which you can't always catch in a sleep study, but obviously, again, having a spouse or someone sleeping with you helps catch that. And probably within 10 years, about 2/3 of people go on to develop a Parkinsonian's disease. A lot of, I think it was really got higher profile as a warning sign when Alan Alda finally came out with having Parkinson's disease, and said that he was diagnosed because he was acting out dreams. So again, more sleep stuff, but once you get to the prodromal stages where people are still mobile, I'm not really seeing as much of a sex-linked bias, except that what Parkinsonian's disease people tend to get might be different, as far as maybe Lewy body versus Parkinsonians versus tremors. Sorry I talked too long. Bye.
No, thank you for that information. You know so much more than me here. So I'm glad that you came to share that with us. Yeah, it's interesting how much sleep, yeah, how much quality of sleep apparently, and how we sleep, um, uh, first of all, maybe the best way of diagnosing things early, and also influence our health for long-term quality of life health so much. So yeah, we should really study it more and focus on that. So thank you for that.
Oh, I also
Just
just
clarify. Did,
oh,
did you
sorry.
say that? Did you say that if you are able to act out your dreams, you're more likely to get this disease? Is that what you said a second ago?
Yes, it is absent any medication that is so normally during REM sleep you basically are in a state where you kind of disconnect your muscle system. If
Sleep
you have
paralysis.
movement during REM sleep, that's what you can catch in sleep studies. Not a great sign. If you act out your dreams absent any um, and again, we don't have any sort of statistics except I just asked my lab mates informally. So this is very informal as far as maybe more from a clinical standpoint, just how people feel about it. Yes, acting out a dream during REM sleep is a really strong indicator of a uh, something that might develop into Parkinsonians down the line, or again might be diagnosable at that point.
So
Because
just
yeah.
to clarify, are you talking about like sleepwalking or having
Oh,
dreams
oh,
that?
oh, no, 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 not sleepwalking. That is very different. No, no, don't worry about sleepwalking. This would be so I don't know if you've ever woken up from a dream and been frozen where you can't move. That is the typical experience.
I
Um,
have done that a couple times, but only maybe
yeah,
once or twice in my whole life.
yeah. Well, I am keeping my fingers crossed that I am on medication that is promoting some amount of REM movement. I have woken myself up by doing a roundhouse kick that I had in my dream and kicked myself out of bed. You know.
Nice.
So yeah, nice stuff like that. So specifically, again, to share Alan Elda's story, he had a dream about eating a marshmallow. He was eating his pillow. Sad to say, I really like Alan Elda.
I like that one.
So so things like that, whether it is a sudden motion that you are doing or something like that, and other things are not going to fall under it. Like I have severe myoclonus, so I'm hoping that what I'm doing is related to that and not pre-Parkinsonians. But yeah, again, the formal diagnosis has to do with movement, but there are a lot of prodromal symptoms. But unfortunately, the biggest ones are constipation and depression, and sometimes loss of smell, which especially these days isn't very indicative anymore. Um, yeah, well, I could go on about Parkinsonians, but I will just say, it looks like there are neuronal and skin changes and or neuronal changes that you can see at the level of the skin. People are looking for that. But um, stepping back, we are not doing a good job of diagnosing everyone with Parkinson's disease, and in particular, poor people and BIPOC people tend to get left out of this. It is we don't even have BIPOC people in our studies, you know?
So
What is
It's
BIPOC
a big
people? Is that
problem, and we talk about it because there's a lack of diversity. We are basically studying white males because, again, the women don't have, even the white women don't have the support to come in.
It does ring a bell, or not ring a bell, but it makes me. I kind of have the same caution about the difference between the man and the woman kind of thing, because there's as far as the chromosomes go, there's so much uh, difference that there's actually more not a black and white picture when it comes to boys and girls in world and biology altogether. That I think that drawing that line sort of negates a lot of good science that could come out of it. I think it goes a little deeper than that. You know what I'm saying?
Uh, true, but in this case, our speaker had a specific hypothesis that it had to do with uh, uh, sex hormones, which it would make sense then to have a difference between men and women based on
Well, isn't there a
hormonal
whole bunch
levels,
of
because
people that
that's
have
the major
kids?
difference.
Isn't am I wrong by saying that there's a whole bunch of people who actually have it's not just two types of chromosomes, and there's a lot of people, there's a lot of males who actually have ovaries, and there's a lot of a blurring of the line versus such a black and white transition. Am I wrong by saying that, or no?
I think that about, and usually it's much more subtle. You might be convolving. Sometimes there are. It's possible to play around a little more with the sex chromosomes, so you can get triple X, XXY, things like that. Um, and something that plays into that doesn't is not necessarily chromosomal is I believe the statistics are about 1 out of every 1,000 births has some intersex characteristics.
It's not directly on the comment, but do you want to like talk about Trisha's case?
I don't need to talk about it, really specifically, but I do have a friend that initially considered herself trans, but then she found out that she had intersex characteristics because again the way her endocrine system was working, and genetically she is XY, but uh, just by who she is, and maybe the influence of having some intersex characteristics, she considers that she was originally intersex, uh, and she was in California, where they're a little more liberal, and she was able to amend her birth certificate to be born female. So, these things are very fuzzy. People think about the most dramatic cases of having having both uh, um, sexual organs, which happens occasionally, but usually it's a lot more subtle.
Yeah, I just wanted to share I used this news article, but there were a bunch of coming out with it was a study coming out that a simple skin test can confirm Parkinson's diagnosis, because there's a lot of misdiagnosis, way too late diagnosis that is happening, because it's really hard to see, to basically prove that this is Parkinson due to the cell death of the dopaminergic cells, or if something else is happening, especially in the beginning of the onset, or maybe hopefully the
Yeah,
test
that's
will maybe help with the earlier. And there was another
yeah,
for
yeah.
Alzheimer's that
Sorry, go on, but yeah, the Yeah, that was what I was mentioning, that one of the one of the very early prodromal might have to do with neuron changes in the skin, which is something you could test for. But as but again, you would have to think about doing this, because you kind of right now we do not have anything that is protective, that we absolutely know or even strongly suspect is protective uh, against the progression of Parkinson's disease. One of the things is catching it at that skin level, is that might be prior to brain changes, because we are thinking that there are changes systemically in the body before it reaches the brain. So kind of uh, there would be hope that we could do more in that case, number one, and or earlier is better, because exercise is our best guess at something that is uh, at least slightly protective. Um, anyway, the where was I going with that? The other thing that is really fascinating is that there is a woman that could smell Parkinson's disease. And this was proven in a double-blind study. She got one wrong, and that was that she identified someone that this is like out of 20, it was 10 and 10. She got one of the controls wrong, but guess what? This person developed diagnosable Parkinsonians 2 or 3 years later. So what's really exciting is that was demonstrated a while ago, but in the past year, uh, and unfortunately it's proprietary, they're not going to tell us much about it, but they did isolate the compound that she is smelling. So that could be really amazing too. But I think, as Katerina and I both know, there's having the idea to do a test, which can be complex, and you have to think about doing it. Parkinsonians is strongly environmental as well, and doesn't necessarily run in families. It depends. And having different means, like well, I'll let Katerina tell you about the motion stuff. I mean, people changing their speech patterns to try and start screening widely could be really amazing too, because again, we're already missing a lot of people.
Yes, we are, and way too late diagnosing. And that is very interesting. You told me that before, and then I looked this up and I found it again, that there is a artificial intelligence is developing a sense of smell. So hopefully they can train artificial intelligence to that smell. That would be interesting, and maybe our phones can tell us if we are developing Parkinson earlier on. That would be quite, or our watches or something would be interesting. Yeah.
If I can remember correctly, didn't I hear something about an animal being able to smell this type of thing, or was that just cancer? I can't quite remember.
Yeah, cancer definitely, but could be also other disease. I'm not sure.
I mean, uh, do people with Parkinson's they also have breathing problems other than the loss of smell?
No, there aren't breathing problems. It is fortunately, it's not affecting the uh, autonomic systems. It has to do with taking out the dopaminergic hind parts of the brain that have all of our patterns for walking and all the redundancies. Um, Parkinsonians patients, you can demonstrate that they are spending conscious effort in trying to walk. uh, you can basically crash their walking ability by asking them to say count backwards in threes from 100. If you have another cognitive task, and that's why coming to a doorway, coming to an elevator, all of these things are challenging because they're trying to process something new, and they are processing moving their limbs.
Yeah, interestingly, such things as like riding a bicycle, sometimes movements like that that are more cerebellum driven, they work just fine. Like all of the, if you give like a stimulator, like if somebody else induces like automized learned movements like these, um, like they have a hard time starting this movement. But once they start, they are very interesting, like videos that a person with Parkinson's can perfectly find. Ride a bicycle. Soon as he steps off the bicycle, then you see the the regular pattern of Parkinson's disease. So it's I think they're probably on YouTube or so. Video.
Yeah. What is really odd, and unfortunately it's hard to get a handle on what to do with this research wise to help or show what's going on. But Parkinsonian's patients, this was actually noticed by a caregiver that uh, her father, I think, was actually really good at walking up and down stairs. So then she had this idea that she was going to put fake stairs in the hallway, and somehow that worked. That's something people aren't formally doing online, is laying down fake stairways for the people they're caring for. And there again, it's the PI in my lab, is very interested in this because we're in the movement disorders side of things, but it's a little hard to figure out what sort of a research project you could do from this because there's so many variables. you know
Well,
anyway.
that's fascinating that
Yeah,
a
but it's
visual cue.
fascinating and formally because again, somehow the stairs are working.
It's like a
Well,
glitch
yeah,
matrix.
yeah, the visual cue. I mean, it provides a visual pathway to sort of, reduce the complexity of the task. At least that's
Well,
fascinating.
we actually have no idea why it works. I mean, that's something we could try doing, is again tasking them and doing all of this stuff. But again, I mean, there are so many variables. I mean, do you have lines on the side? Is it just the stairs? Is it the how far apart are they? Things like that. It starts to get difficult to even parse what is working. Is basically unfortunately, and there are and it's a little off what we are doing, which again is we are trying to catch what movement disorders are going on in the pre-Parkinsonian stage with the sleep disorders, and then at the very early stage, along with regular work
I think
in the typical Parkinsonian.
I think interesting would be if technology advances more once one knows the onset of Parkinson to just let the cerebellum take over more and more behaviors and just have a trigger and in the striatum, like a triggering electrode, a tiny one in the striatum to start the movement and let the cerebellum take over the movement, and then having a stop signal whenever the person wants to stop, like to self trigger, or it combined with some augmented reality or something to have like a smart loop that basically triggers a movement while the dopamine cells are dying, train the cerebellum to take over more movement behaviors. And the only thing the device then has to do in the end is just triggering the striatal neurons, and then give a stop signal when to stop the movement. But this would involve a lot of like new technology and like the person would need that trained person by person, so it would be really expensive, I guess, and quite elaborate.
Well, yeah, we have some less fancy stuff, but that is interesting because unfortunately, what my lab mate just proved a couple years ago. So we know really well that external triggers um, help people in general to have better step initiation, like when you're just beginning to move. That's what I'm looking at. Except for Eli, don't let him be part of your study. I mean, okay. Um, he does worse if you give him a cue. He crashed my data. So, like, don't let people you know in your study. Okay. Um.
Well, we all
Anyway,
know and
so
appreciate and
well,
love
actually,
Eli as a special case.
this no no this did lead to something that was interesting. That I have been uh uh making myself not even informally look into, because there's one other elderly person that did worse with a Q, and there are maybe um maybe about 5% of the Parkinsonians patients, maybe about four, didn't do as well with the Q either. But anyway, what is really unfortunate is self queuing doesn't work. It has to be an external cue. So what people are looking at is, especially when people freeze, to have some sort of a cue, whether it's a noise or something that goes to their phone or something like that helps them get unstuck, because you will. Some people freeze with Parkinsonians. You get to the stage where you just freeze and you can't move. It's like molasses. So now they're trying to figure out getting AIs to figure out when people are stopped because they want to stop, and when they are stuck. So yeah, we have our good freezers, and we're putting them through an obstacle course of like an elevator door and all this other stuff to freeze at, and then sometimes they get to stop when they want to. So but yeah, so even just a noise or some sort of a cue is helps everyone step off, and Parkinsonians a bit less than other people, but way better than trying to do it on their own. But yeah, the so how what would there be other ways to access the cerebellum externally? Katerina, do
Uh,
you know?
that's a good question. I think, yeah, it's interesting. I have to think better about how to basically make maybe a direct connection, like,
Yeah,
or a better or increased connection between the
mhm,
striatal projection neurons
mhm.
and the cerebellum without having to have this dopamine release somehow.
Okay.
Maybe
What about could you do it with big
I don't
magnets?
really, yeah, yeah,
Target?
that was,
Would that work?
yeah, I was thinking that somehow, or having different,
Mm,
especially the training, right? To
yeah.
so for example, yeah, the cerebellum can learn quite quite complex
Yeah.
behaviors. Like bicycle riding is not.
It does. It's fascinating.
Yeah, so the question is, how can we use that and use that more and more? Like, drive away, drive the, like, make the cerebellum take over more and more responsibilities while Parkinson is, yeah, the disease progresses more and more, basically. I don't know. One would have to look into how to do that, but I would say to somehow record while people still have like relatively okay movement patterns, to record those patterns, like the
Mm-hmm.
activity patterns, and then
Mm-hmm.
constantly uh, give that as a stimulus to the cerebellum, so they
Mm-hmm.
hopefully um, would take over that memory, basically.
Hmm.
That was the idea I had so far.
Okay.
And then how to trigger, and then somehow trigger those activity patterns that one, gave them as an input, and then recall them somehow. So maybe an implant or so that would, over the years, give, that make the cerebellum learn all those behavioral patterns,
Hmm.
and then one would just need maybe to trigger us in a specific way, like now walk and now do this and that. So yeah, so basically train the cerebellum to do all the movements instead of just a few. um yeah, would
But
be an
just
interesting
but just
study.
stepping back, do you think you could try some of that out by stimulating the cerebellum with TIMS just to see if you can the transcranial magnetic stimulator?
Yeah, it would be interesting to try it out. i think I think that would be really interesting. I don't know if somebody is working on this, but I think it would be
Hmm.
really cool.
Hmm. Yeah, because there's one in the department. We're using it in a different research study. So,
Yeah, pitch. I
but
think.
that's not something I'm really very uh, involved with.
Yeah,
Yeah.
I was thinking of the implants because there's this paper that came out where they created the chip, and they
Hmm.
recorded basically if for paralyzed people, so they recorded.
Yeah.
Yeah,
Yes.
and then,
Yeah.
you know so one could either try this to put the
Mm-hmm.
chip and like totally leave the brain out, basically. I think Miguel Nicolelis is working on something like that, so define a stimulation for Parkinson, or just do the same but with the cerebellum, but one could
Mm-hmm.
first maybe try with the magnetic stimulation.
Yeah, just for proof of concept, because when they put in because I mean right now the big thing is which once when do you put in, they do have brain implants where they're stimulating parts of the brain, but it's not the cerebellum. So right now it's deciding the trade-offs between the two that they're doing, where to target it better, what patterns to do. I mean, fortunately, you can program in whatever patterns you want, but and then but it's based on basically the technology that you have, and different people need different patterns, and there's no sense to it. There's no sense to even people walking, which is annoying.
Yeah.
So actually, like going back to the topic of cueing people or shaking people out of their, I shouldn't say shaking, providing a stimulus that gets people out of being frozen. Has there been any attempt to do like the really basic stupid smartphone app that like just detects that it's not moving around and emits a ping?
Yeah, I don't know any of this. Leslie, do you?
Leslie?
I'm not sure. Maybe you muted.
Or yeah, you're muted, Leslie.
I think that's a really good idea, Eli. One should try that. I don't know if anyone is trying it, or give this little vibration things on the iWatch. That would maybe also be. I know that there's haptic technology, but I think it's more in the terms of getting signals that you wouldn't hear to your skin. But that would be interesting if that would be enough to unfreeze somebody. I'm not sure.
If we've lost Leslie, I will take that up with her later.
Okay. Yeah, it's getting quite late here. A really early morning tomorrow. So thank you very much for coming and asking all these interesting questions, and Leslie for sharing so much knowledge about the Parkinson studies. And so thank you all very much. So follow the Science Society if you enjoy discussions like these, and we have more guest speakers coming. So tomorrow will be a really cool room about how spiders fly. It will be really interesting, about ballooning spiders. Has to do with physics, and it's I really thought this was really cool. So, and both like the first author and the last author of the paper will be coming. Then Dr. Singupta from Yale University will be talking about her latest research on how neurons find their place. It's a developmental study. It's really interesting and also really important especially for autism and so on, because there has been shown that neurons have an issue during development finding the right place. And then on Wednesday we have Dr. Stock and also his PhD students coming to present the largest psychedelic study, 6,000 or so. Anyways, on trips and neurotransmitters. So this will be really interesting to hear about that study. And then on Thursday we have Dr. Liu talk. If I don't remember wrong, I think he's at Penn State. Yes, he's at Penn State, professor. He developed a new theory of entropy to solve a lot of material design problems that will lead us to a lot of new cool technology. So, and also just the basic science part of it is really cool. And yeah, so join us again. They will be all at this time at 9 p.m. EST this week. Yeah, thanks for coming, and hear you again soon, hopefully.
Thanks, everyone. Great room.
Okay, we'll close the room, and 3, 2, 1. Bye, everyone.